Hello everyone, my name is Humberto Sotomayor. I graduated from FIU in Business Administration with a major in Information Technology. When my first son was born, he was diagnosed with autism when he was 3, which was known to be a condition transmitted genetically. In this crisis, I sought the advice of my father, who is a medical doctor of anesthesiology in Mexico, and I knew he would direct me to the best specialty physicians in obtaining the best medical care needed for my son. This is where passion in researching the latest information of my son's health began. In 2003, my second son was born, and 3.5 years later he was diagnosed with muscular dystrophy. It is also known to be a condition transmitted genetically. This situation changed the path of my life, and since then I have been researching health to help my kids to have a better quality of life, trying as much as possible to reduce the progression of their illnesses. I have visited and consulted with several naturopath doctors over the last 6 years, and I have attended several health seminars across the United States. I have read many books, and although I haven't been able to find a cure for my son's conditions, I have been exposed to several health protocols that allow for anyone to achieve homeostasis in their lives. I know that following these protocols, I have at least been able to reduce the progression of my kids' illnesses. Most sicknesses our populations are facing are induced by lifestyle choices overwhelming our systems with toxins and free radicals, and it is our responsibility to learn and act upon, especially because we are bombarded every day with unhealthy food choices. It is my wish to share with you this presentation, where scientific studies have demonstrated that oxidative stress and mitochondrial dysfunction are linked to many degenerative diseases. I also would like to state that although I'm not a physician, having a father as a physician and several extended family members and friends that are also in the medical and wellness and healthcare professions, they have helped me and mentored me along with the research that I continue to seek for both my son's medical conditions. In this presentation, I want to talk to you about oxidative stress, mitochondrial dysfunction, premature aging, metabolic syndrome, and the correlation among them. I'm also going to talk about antioxidants and nanotechnology. Some mitochondrial dysfunctions are known to be genetic, but many others are lifestyle related, and where oxidative stress is present and leads to premature aging and to over 250 diseases, including metabolic syndrome. Free radicals are also known as reactive oxygen species, and they are molecules that are missing an electron in their outer orbit, and therefore they are unstable and cause damage to mitochondrial DNA as well to nuclear DNA. Antioxidants is our line of defense to neutralize free radicals, and they are produced by mitochondria within the cells. However, antioxidants are also present in many natural food products and can help our bodies with this defense mechanism. Let's start by explaining what the role of mitochondria is in our body. Mitochondria are intracellular organelles that play a crucial role in energy metabolism. They generate this energy as they breathe in oxygen and are able to metabolize glucose effectively. As this is happening, oxidation also occurs, creating free radicals, which are unstable oxygen molecules. These free radicals, when are not neutralized in an effective way, they impair our mitochondrial metabolism and damage the mitochondrial DNA and nuclear DNA. However, our body has the ability to fight this oxidation through protective enzymes that neutralize free radicals. These enzymes are best known as antioxidants because it's what they do. They prevent oxidation by neutralizing free radicals. These enzymes are also known as superoxide dismutase and glutathione peroxidase and catalase. These enzymes have the capacity to donate an electron to these free radicals and neutralize them. When this happens, water and oxygen are produced within the cell, and the cycle is completed without any damage to our DNA. Mitochondria are responsible for creating more than 90% of the energy needed by our body to sustain life and support growth. When they fail, less and less energy is generated within the cell. Cell injury and even cell death follow. Diseases of the mitochondria appear to cause the most damage to our cells of the brain, heart, liver, skeletal muscles, kidney, and the endocrine and respiratory systems. Mitochondria are also involved in other functions tied to the specialized duties of the differentiated cell in which they reside. These are intimately involved with most of the major metabolic pathways used by a cell to build, break down, and recycle its molecular building blocks. They are required to make hemoglobin. In the liver, mitochondria specialize to detoxify ammonia in the urea cycle. Mitochondria are also required for cholesterol metabolism, for estrogen and testosterone synthesis, for neurotransmitter metabolism, and for free radical production and detoxification. They do all this in addition to breaking down the fat, protein, and carbohydrates we eat and drink. Many rare metabolic, neurodegenerative, and metabolic disorders have been associated with mitochondrial dysfunction, and these include metabolic syndrome, diabetes, cardiovascular disease, Parkinson's disease, Alzheimer's disease, Huntington's disease, amyotrophic lateral sclerosis, better known as ALS, fibromyalgia, chronic fatigue syndrome, among many others. Energy metabolism impairment and oxidative stress are common in all of these diseases. In fact, oxidative stress is present in over 250 diseases and seems to be the #1 cause of mitochondrial dysfunction when it's not genetic. Oxidative stress is lifestyle related, and it leads to all sorts of complications depending on which cells are being affected. Oxidative stress may lead to cancer, diabetes, metabolic syndrome, and many neurodegenerative diseases and cardiovascular disease, which all cause premature death. Other external factors of free radicals are environmental contamination, excess weight, toxic habits, stress, poor nutritional knowledge, prolonged sun exposure, and already ongoing diseases like diabetes, arthritis, Crohn's disease, AIDS, damage to the central nervous system, cancer, hepatitis, chronic kidney failure, cardiovascular disease, and asthma. This diagram is from the American Heart Association, and it shows an increase of reactive oxygen species, which creates a free fatty acid overload and hyperglycemia, which cause an increase on cell mutations, a decrease in mitochondrial biogenesis, leading to aging, polymorphism, and mitochondrial dysfunction, causing lower energy levels and to an increasing number of reactive oxygen species, creating a biochemical chain reaction that leads to insulin resistance, diabetes, and cardiovascular disease. This is a slide from the Journal of Cell Science, and it shows how an imbalance where there is more cellular oxidative stress than cellular stress defense, and how it damages mitochondrial DNA. This causes an increase of reactive oxygen species, leading to a mitochondrial crisis, to cellular death, causing premature aging. Now, to show evidence of the relationship between oxidative stress with mitochondrial dysfunction, metabolic syndrome, and other diseases, in this section I'm going to show research that has been published in PubMed.org, which is the United States Medical Library, and I'm just going to highlight briefly in each one of them. And this first one reads, many lines of evidence suggest that mitochondria have a central role in aging-related neurodegenerative diseases. Mutations in mitochondrial DNA and oxidative stress both contribute to aging, which is the greatest risk factor for neurodegenerative diseases. This other article is about mitochondrial dysfunction and oxidative stress in aging and neurodegenerative diseases, and it reads as follows. It says, a major risk factor for neurodegenerative diseases such as Parkinson's disease, Huntington's disease, amyotrophic lateral sclerosis, Alzheimer's disease, and progressive supranuclear palsy is aging. Two processes that have been implicated in aging are free radical-induced oxidative damage and mitochondrial dysfunction. And it concludes, altogether, the age-dependent onset of the progressive course of the neurodegenerative diseases may ultimately highlight an association between aging, mitochondrial impairment, and oxidative stress. This other article is titled Mitochondria, Oxidative Stress, and Neurodegeneration. It says as follows. The central nervous system functions depend heavily on efficient mitochondrial function, since brain tissue has a high energy demand. Mutations in mitochondrial DNA, generation and presence of reactive oxygen species, and environmental factors may contribute to energy failure and lead to neurodegenerative diseases. Mitochondria have an important role in aging-related neurodegenerative disorders. This is another article by the American Physiological Society, and it reads as follows. It says, several lines of evidence indicate that mitochondrial dysfunction as a major contributor to cardiovascular senescence. Besides being less bioenergetically efficient, damaged mitochondria also produce increased amounts of reactive oxygen species, with detrimental structural and functional consequences for the cardiovascular system. The age-related accumulation of dysfunctional mitochondria likely results from the combination of impaired clearance of damaged organelles by autophagy and inadequate replenishment of the cellular mitochondrial pool by mitochondriogenesis. This other article says mitochondrial dysfunction and cancer metastasis. It says as follows. It says mitochondria have an essential role in powering cells by generating ATP. They are also the major source of generating reactive oxygen species, which have regulatory roles in cell death and proliferation. Mutations in mitochondrial DNA and dysregulation of mitochondrial metabolism have been frequently described in human tumors. Although the role of oxidative stress as the consequence of mutations and/or other altered mitochondrial functions has been demonstrated in cancerogenesis, a causative role of mitochondria in tumor progression has only been demonstrated recently. Specifically, the subject of this mini-review focuses on the role of mitochondria in promoting cancer metastasis. Cancer relapse and the subsequent spreading of cancer cells to distal sites are the leading causes of morbidity and mortality in cancer patients. Recently, it was demonstrated that mitochondrial oxidative stresses could actively promote tumor progression and increase the metastatic potential of cancer cells. This is another article published in Lance Bioscience. The title of it is Oxidative Stress in Cancer-Associated Fibroblast Drives Tumor Stroma Coevolution. And it reads as follows. It says, we provide evidence that oxidative stress in cancer-associated fibroblasts is sufficient to induce genomic instability in adjacent cancer cells via a bystander effect, potentially increasing their aggressive behavior. Now let's jump into metabolic syndrome, and let's start by defining it. Metabolic syndrome, so we can have a better understanding, it's a cluster of conditions that occur together in a same person, increasing the risk of heart disease, stroke, and diabetes. And this cluster of conditions are increased blood pressure, high blood sugar levels, excess body fat around the waist, and abnormal cholesterol levels. Other names for metabolic syndrome are also metabolic syndrome X, cardiometabolic syndrome, syndrome X, insulin resistance syndrome, Raven's syndrome, and in Australia they know it as COWS. This article is very useful because it provides the biomarkers found in metabolic syndrome. So after this slide we will see the graphs related to this study, and the article reads, previous evidence supports the important role that oxidative stress plays in metabolic syndrome related manifestations. In this study done in close to 100 people, they determined the relationship between the number of metabolic syndrome components and the degree of oxidative stress in metabolic syndrome patients. Participants were divided into 4 groups based on the number of metabolic syndrome components. So I'm going to show you the results in the following 3 slides, which will give us a better understanding of the increasing risks. You can see here at the top chart A, which is related to the ischemic reactive hyperemia, and this refers to blood flow to the brain. And you can see as there's more metabolic syndrome components present, the blood flow to the brain goes down, and this could lead to a risk of neurodegenerative diseases or reduced cognitive functions. On chart B in the middle, this is related to inflammation in the coronary arteries, and you can see the increased inflammation as there's more metabolic syndrome components present, and this could lead to a risk of cardiovascular disease and stroke. On chart C of the bottom of the screen, it is related to the total nitrite levels in the plasma, and reduced nitric oxide availability. It's a hallmark of endothelial dysfunction, and you can see a decline here as there's more metabolic syndrome components present. On this page, on the chart at the top, it's related to hydrogen peroxide levels, and this is related to the cardiovascular disease and to higher free radical activity. As you can see in the chart, as there's more metabolic syndrome components present, it is increasing. On the chart at the bottom, this is related to lipid peroxidation levels, which refers to the oxidative degradation of lipids, and you can see it increases as there's more components of metabolic syndrome present. By the way, lipid peroxidation is the most common biomarker of oxidative stress. This slide refers to the antioxidant activity of superoxide dismutase and glutathione peroxidase respectively, and you can see that they both increase as there's more components of metabolic syndrome present. The top charts, in the case of SOD, activity decreases when the 5th component of metabolic syndrome. In this section of the presentation, we will continue to see research from PubMed, but I will focus on studies done in 3 main antioxidants. First, we're going to look at rosemary and all of its constituents, like carnosic acid, rosmanol, carnosol, rosmarinic acid, galdosol, ursolic acid. Then we'll jump over to olive leaf extract, and lastly we'll get into anthocyanins and proanthocyanins derived from the black bean. This study is about Rosmarinus officialis, which is the scientific name for rosemary, and it says that its extract suppresses the Propionibacterium acnes induced inflammatory responses, and it says that Propionibacterium acnes is a key pathogen involved in the progression of acne inflammation. In this study, it was investigated the inhibitory effect of rosemary extract on induced acne inflammation in vitro and in vivo. The results showed that rosemary extract significantly suppressed the secretion and the pro-inflammatory expression of the cells. This study is about the protective effects of carnosic acid in light-induced retinal degeneration. The herb rosemary has been reported to have antioxidant and anti-inflammatory activity. Here we investigated the antioxidant and neuroprotective activity of carnosic acid in retinal cell lines exposed to oxidative stress and in a rat model of light-induced retinal degeneration. Retina-derived cell lines treated with hydrogen peroxide were used in vitro models for testing the protective activity of carnosic acid, and for in vivo testing, dark-adapted rats were injected carnosic acid prior to exposure to white light to assess protection of the photoreceptor cells. In vitro, carnosic acid significantly protected retina-derived cell lines against hydrogen peroxide-induced toxicity. And similarly, we found that carnosic acid protected retinas in vivo from light-induced retinal degeneration. These findings suggest that carnosic acid may potentially have clinical application to diseases affecting the outer retina, including age-related macular degeneration and retinitis pigmentosa, in which oxidative stress is thought to contribute to disease progression. This slide is about the rosemary constituents that have anti-arteriosclerotic effects, and it reads as follows. Arteriosclerosis is a chronic and progressive inflammatory disease. Novel anti-inflammatory therapies may have promise as treatment strategies for cardiovascular risk reduction. Rosemary has been used in folk medicine to treat headaches, epilepsy, poor circulation, and many other ailments. It was found that rosemary could act as a stimulant and mild analgesic to reduce inflammation. This study investigated effects of rosemary components, carnosic acid and carnosol, on cell migration, and it concludes, rosemary, especially its carnosic acid component, has potential anti-arteriosclerotic effects related to cell migration. This slide is about the neuroprotective effects of carnosic acid, and it says that ischemia and hypoxia reduce oxidative stress, which is associated with neurodegenerative diseases. The present study investigated protective mechanism of carnosic acid on ischemia and hypoxia-induced neuronal cell injury. The results showed that carnosic acid reduced 52% of the infract volume from brains under ischemia in vivo, and it protected the neuronal cells from hypoxic injury in vitro. And it concludes, carnosic acid protected neuronal cells under ischemia and hypoxia through scavenging or reducing the oxidative oxygen species and nitric oxide by its anti-inflammatory and antioxidative properties. This study is about the anti-inflammatory properties of carnosic acid in skin, and it says carnosic acid is a diterpene compound exhibiting antioxidative, anti-cancer, anti-angiogenic, anti-inflammatory, anti-metabolic disorder, and hepatoprotective and neuroprotective activities. Therefore, our data strongly suggest the potential of carnosic acid as an anti-inflammatory drug against skin inflammatory responses with inhibitory properties. This is a study where carnosic acid prevented induced cancer in hamsters. And it says that 2 groups of hamsters were induced cancer in their buccal pouches by painting with a cancerogenic laboratory substance 3 times a week for 14 weeks. While one group developed oral tumors, 100% a well-differentiated carcinoma, and it was accompanied by deregulation of molecular markers was noticed also in the hamsters. The other group, oral administration of carnosic acid to the second group, not only did it completely prevented the tumor formation, but it also corrected abnormalities in the expression pattern of molecular markers. This study suggests that carnosic acid might have inhibited the tumor formation by exerting anti-cell proliferative, anti-inflammatory, anti-angiogenic, and apoptotic potential. This is a study related to weight gain and how rosemary leaf extract limits weight gain. The study says rosemary extracts are natural antioxidants that are used in food supplements, and cosmetic applications. They exert anti-inflammatory and anti-hyperglycemic effects and promote weight loss. which can be exploited to develop new preventative strategies against metabolic disorders. Therefore, the aim of the present study was to evaluate the preventive effects of rosemary leaf extract that was standardized to 20% carnosic acid on weight gain, glucose levels, and lipid homeostasis in mice that had begun a high-fat diet as juveniles. The mice were broken down into three groups. Some animals were given a low-fat diet, some others were given a high-fat diet, and the third group was given a high-fat diet that was supplemented with rosemary extract. Physiological and biochemical parameters were monitored for 16 weeks. Body and epididymial fat weight in animals on the high-fat diet that was supplemented with the rosemary extract increased 69 and 79% less than those in the high-fat diet group. Treatment with rosemary extract was associated with increased fecal fat excretion, but not with decreased food intake. The extract also reduced fasting glycemia and plasma cholesterol levels. In addition, they evaluated the inhibitory effects of rosemary extract in vitro on pancreatic cells, which regulate fatty acid storage and glucose metabolism. The in vitro findings were correlated with the observations in the animal experiments. Thus, the present results suggest that rosemary extracts, that is rich in carnosic acid, can be used as a preventive treatment against metabolic disorders. This is a study that shows the anti-cancer and anti-inflammatory potency of rosemary. And it says that the supercritical fluid treatment of rosemary leaves, in which 5 major active principles were identified, which are rosmarinic acid, carnosol, 12-methoxycarnosic acid, carnosic acid, and methyl carnosate. The extract showed phenolic content equivalent to gallic acid. It showed high scavenging scores of DPPH, which is a radical used in laboratories, of 82%. Nitric oxide inhibition occurred in mouse leukemic cells. The extract inhibited dose-dependent viability suppression and significant tumor necrosis factor alpha production in human hematoma cells. Conclusively, rosemary can be considered an herbal anti-inflammatory and anti-tumor agent. This is a study about the radioprotective and anti-mutagenic effects of rosemary. And it says that the radioprotective effects of carnosic acid, carnosol, and rosmarinic acid against chromosomal damage induced by gamma rays compares with those of L-absorbic acid and dimethyl sulfoxide. Carnosic acid and carnosol are the only compounds that showed a significant anti-mutagenic activity both before and after gamma irradiation treatments. This is a study regarding rosemary and cancer prevention. And it says colorectal cancer is the second leading cause of cancer death in Australia. Nutrition, particularly the intake of vegetables and certain plant components, has been reported to have a major role in cancer risk reduction. Recently, there has been a growing research interest in rosemary, a common household plant grown in many parts of the world. This study aims to review scientific evidence from all studies published from 1996 to March of 2010 that examined the protective effects of rosemary on colorectal cancer and other types of cancer. Literature evidence from animal and cell culture studies demonstrates the cancer potential of rosemary extract, carnosol, carnosic acid, ursolic acid, and rosmarinic acid. The reported anti-cancer properties were found to arise through the molecular changes in the multiple stage process of cancer development. This is evidenced by the ability of rosemary to suppress the development of tumors in several organs, including the colon, breast, liver, stomach, as well as melanoma and leukemia cells. The results suggest that the different molecular targets modulated by rosemary and its active constituents are useful indicators of success in clinical cancer chemoprevention trials. Now I am going to share with you why rosemary is so effective. On the diagram to the left, we illustrate how an antioxidant like vitamin E works. This antioxidant shares one electron to neutralize a free radical, and then it deactivates. In the diagram to the right, we illustrate how carnosic acid from rosemary shares one electron to the free radical, and it's neutralized. Carnosic acid is metabolized and reactivated, creating carnosol. Carnosol then shares one electron to a second free radical, and it's neutralized. Carnosol is then metabolized and reactivated, creating rosmanol. Rosmanol then shares one electron to a third free radical, and it's neutralized. Lastly, rosmanol is metabolized and reactivated, creating galdosol. Galdosol then shares one electron to a fourth free radical, and it's neutralized. Now I'm going to jump over to our olive leaf extract research, and this first one is about olive leaf extract and arthritis. And the first study reads, the olive leaf extract has antioxidant and anti-inflammatory actions. However, the role of olive leaf extract in mechanical inflammatory arthritis or osteoarthritis is unclear. This study investigated the olive leaf extract on the development of induced arthritis. In mice, it significantly ameliorated paw swelling, and it suppressed induced ear edema. In the human monocyte cell line derived from leukemia patients, it reduced the tumor necrosis alpha. These results indicated that olive leaf extract is an effective anti-arthritis agent through an anti-inflammation mechanism. Also, olive leaf extract may be beneficial for the treatment of osteoarthritis in humans. This is a study on olive leaf extract and metabolic syndrome. This study was done on insulin sensitivity in middle-aged overweight men. And it says olive plants have been used for centuries in folk medicine to treat diabetes, but there are very limited data examining the effects of olive polyphenols on glucose homeostasis in humans. The objective of this study is to assess the effects of supplementation with olive leaf polyphenols on insulin action and cardiovascular risk factors in middle-aged overweight men. The study was done with 46 participants, ages 40 to 52 years old, in a randomized, double-blind, placebo-controlled crossover trial in New Zealand. The results showed that olive leaf extract supplementation was associated with a 15% improvement in insulin sensitivity compared to placebo. There was also a 28% improvement in pancreatic beta cells responsiveness. The study concludes supplementation with olive leaf polyphenols for 12 weeks significantly improved insulin sensitivity and pancreatic beta cell secretory capacity in overweight middle-aged men at a risk of developing metabolic syndrome. This is a study on olive leaf extract, anti-arteriosclerotic, and suppressed inflammation. And it says olive leaf extract is related to suppressed inflammatory response in rabbits with experimental arteriosclerosis. The anti-arteriogenic effect of olive leaf extract is supposed to be related to its activities of antioxidation and anti-inflammation. Rabbits were assigned to three separate groups. One which was a control group that was fed a regular diet. The second group was fed with a high lipid diet, and the third group was fed with a high lipid diet but supplemented with olive leaf extract. The control group serum levels were normal, but the high lipid diet supplemented with olive leaf extract group showed significantly lower inflammation, cholesterol, triglycerides, and low density level cholesterol levels than the high lipid diet group. This study proved the effect of olive leaf extract on inhibition of arteriosclerosis, which is related to the suppressed inflammatory response. This is a study in which olive leaf extract was compared to other antioxidants in fighting oxidative stress. And it says that modulatory effects of resveratrol and citrofablan and plant-derived extracts on oxidative stress in human leukemic monocyte lymphoma cell line. The objective of this study was to determine the concentration of compound that inhibited cell growth by 50%. Grapeseed, polyphenols, olive leaf extract, bearberry, and echinacea were examined. Cellular reduced glutathione levels were measured as an indicator of oxidative stress. DNA damage was also assessed. And it concludes that resveratrol demonstrated the highest inhibition cell growth, but that olive leaf extract and bearberry strongly reduced the induced oxidative stress. This is a study done in olive leaf extract, and it was compared to captopril, which is a drug on the market since 1976. This is a double-blind, randomized, parallel, and active controlled clinical study that was conducted to evaluate the anti-hypertensive effect as well as the tolerability of olive leaf extract in comparison with captopril in patients with stage 1 hypertension. Additionally, this study also investigated the hypolipidemic effects of olive leaf extract in such patients. After 8 weeks of treatment, both groups experienced a significant reduction of systolic blood pressure as well as the diastolic blood pressure. Such reductions were not significantly different between the groups. However, a significant reduction of triglyceride levels was observed in the olive group, but not in the captopril group. Now we're going to jump over to the black bean properties. In this study, we analyzed the phytochemicals of black beans to evaluate their anti-proliferative and antioxidative activities. Bioactivity-guided fractionation of black bean seed coats was used to determine the chemical identity of bioactive constituents, which showed potent anti-proliferative and antioxidative activities. 24 compounds, including 12 triterpenoids, 7 flavonoids, and 5 other phytochemicals, were isolated. They evaluated the anti-proliferative activities of isolated compounds against human colon cancer cells, human liver cancer cells, and human breast cancer cells. 11 compounds showed potent inhibitory activities against the proliferation of liver cancer cells. 14 compounds showed a potent anti-proliferative activities against colon cancer cell growth. 7 compounds showed potent anti-proliferative activities against breast cancer cell growth, and 6 flavonoids showed potent antioxidant activity. These results show the phytochemical extracts of black bean seed coats have potent antioxidant and anti-proliferative activities. This is a study that shows the protective effect of black beans in diabetes. It says that cooked common beans protects against beta cell damage in streptococcus-induced diabetic rats. Diabetes is a disease characterized by hyperglycemic state that leads into a chronic inflammatory state. They evaluated in vivo effect of a diet supplemented with 25% cooked black bean cultivar negro flour in streptococcus-induced diabetic rats. The effect was assessed before and after the onset of the diabetes. There is a significant decrease in the total phenolic thionins and anthocyanins content after cooking, but the concentration of most of the single phenols analyzed are only slightly decreased. The treatment group showed a significant reduction of glucose of 22.8%, triglycerides by 21.9%, total cholesterol of 29.9%, and the low density level cholesterol by 56.1%, that correlates with the protection of the pancreatic beta cells. The preventive treatment group did not inhibit the tumor necrosis factor alpha and the interleukin 1 beta which is a regulator to inflammation response and to the immune system, whereas the treatment group did, compared to the diabetic control. Therefore, bean supplementation can be recommended to control diabetes. This is a study regarding colon cancer and anthocyanins, and this is a study which was done on rats, and it says anthocyanin-rich extracts inhibit multiple biomarkers of colon cancer in rats. The aim of the present study was to investigate the chemoprotective activity of anthocyanin-rich extracts, and the results support our previous in vitro studies suggesting a protective role of anthocyanin-rich extracts in colon cancerogenesis, and indicate multiple mechanisms of action. This is a study where they compare 4 varieties of beans and their anthocyanin content. They're comparing 4 bean types, among which are mung bean, moth bean, black and red varieties of the Atsuki beans. They were all evaluated for the phenolic content, antioxidant activity, and inhibitory properties against alpha-glucosidase and pancreatic lipase. Results showed that Atsuki bean varieties contain a higher phenolic indexes. Black bean variety was found to be the most active and superoxide anion scavenger. The inhibitory activities of all the extract against lipase were found to be more potent than the alpha-glucosidase, but higher even in the black bean. They all showed strong inhibitory activities on lipase. These results suggest that beans are a potential source of antioxidant phenolics and also great sources of strong natural inhibitors of alpha-glucosidase and lipase activities. They are also good functional food ingredients for promoting health and wellness, and good sources of phenolic antioxidants and strong natural inhibitors of enzymes associated with diabetes and obesity. Anti-radical capacity of black beans. The Hampa bean is a black variety rich in condensed thionins, anthocyanins, and flavonols with interesting biological activities. The objective of this work was to evaluate the anti-radical capacity of Hampa bean methanolic extract and some of the pro-anthocyanin-rich fractions derived from it. The results suggest that black Hampa bean could be an important source of polyphenolic compounds with potential biological use as an antioxidant and anti-cancer agents. Anthocyanins and metabolic syndrome. Obesity is defined as the accumulation of excess adipose tissue resulting from various metabolic disorders. Adipocyte dysfunction is strongly associated with the development of obesity and insulin resistance. Abdominal obesity and adipocyte dysfunction play an important role in the development of metabolic syndrome. This study shows that anthocyanins have a significant potency and anti-obesity and ameliorate adipocyte function in vitro and in vivo systems, and also that they have important implications of preventing metabolic syndrome. This is a study regarding anthocyanins and how they inhibit carbohydrates and sugar absorption. Effects of polyphenols and sugar absorption. Excessive postprandial glucose excursions are a risk factor for developing diabetes associated with impaired glucose tolerance. Postprandial means after eating. One way to limit the excursion is to inhibit the activity of digestive enzymes for glucose production and for the transporters responsible for glucose absorption. Flavonols and proanthocyanidins inhibit alpha amylase activity. Anthocyanins inhibit maltase. Sucrase is less strongly inhibited, but anthocyanidins seem to be somewhat effective. This data is supported by oral glucose tolerant tests on animals and by a limited number of human intervention studies on polyphenol-rich foods. Acarbose is a drug whose mechanism of action is through the inhibition of alpha amylase and alpha glucosidase, and in intervention studies it gives a 6% reduction in diabetes risk over 3 years. And it concludes that a lifetime intake of dietary polyphenols, assuming the same mechanism, has therefore a comparable potential to reduce diabetes
diabetes risk. But more in vivo studies are required to fully test the effect of modulating postprandial blood glucose in humans. This is a study in which type 2 diabetes and anthocyanins are being associated, and the title says dietary flavonoid intakes and risk of type 2 diabetes in US men and women. Data from mechanistic studies support the beneficial effect of specific flavonoids on insulin sensitivity and type 2 diabetes. The objective of this study was to evaluate whether dietary intakes of major flavonoid subclasses, including anthocyanins, were associated with the risk of type 2 diabetes in US adults. And the study concludes that higher intakes of anthocyanins were significantly associated with a lower risk of type 2 diabetes. This is a study regarding microencapsulation of anthocyanins. Anthocyanins are sensitive to the environmental conditions. Thus, their bioavailability in the gastrointestinal tract is an important determinant of their in vivo activity. In this study, the potential benefits of encapsulating an anthocyanin-rich bilberry extract on anthocyanin stability was investigated. Non-encapsulated bilberry extract and 3 other extracts loaded in microcapsule systems were incubated in simulated gastric fluid and simulated intestinal fluid. After exposure to these media, released anthocyanins were identified and quantified. Encapsulation of anthocyanins doubled the amount of available anthocyanins after 150 minutes of incubation. These results illustrate the ability of encapsulation to inhibit early degradation of anthocyanins in the intestinal system. Biomedical applications of nanoantioxidants. For centuries now, antioxidants have been known to provide better health by neutralizing free radicals which are continuously produced in the human body. In normal circumstances, self-antioxidant defense systems of the human body is capable of quantitatively managing the free radicals. However, in certain cases, which are at the threshold of developing diseases like diabetes and Alzheimer's, the human body calls for an external source of antioxidants. Since orally delivered antioxidants are easily destroyed by acids and enzymes present in the human system, only a small portion of what is consumed actually gets absorbed. Hence, there is a recognized and urgent need to develop effective methods for efficiently delivering antioxidants to the required sites. Nanoencapsulation has emerged as a key and efficient delivery process. Designed as a sponge-like polymer, nanoencapsulated antioxidants provide a protective vehicle which keeps antioxidants from being destroyed in the human gut and ensures better absorption in the digestive tract. In fact, the nanocapsules bind themselves to the intestinal walls and pour antioxidants directly into the intestinal cells, which allow them to be absorbed directly into the bloodstream. The use of these highly branched nanometer-sized polymeric materials as nanoantioxidants for prevention and treatment of human diseases associated with oxidative stress is of immense public health relevance globally. This is a comparison of nanoencapsulation of curcumin. In this study, they compared nanoencapsulation of curcumin to non-encapsulated curcumin as cancer therapy. Curcumin is lipophilic, and thus, following oral administration, it hardly appears in the blood, indicating its potential therapeutic challenge in cancer therapy. And it concludes that nanoencapsulated curcumin was found to be a potential formulation in oral route, whereas an identical amount of free curcumin treatment was found to be almost ineffective. In this section of the presentation, I'm going to introduce you to a nanoencapsulation method that was developed by a Franco-Japanese doctor by the name of Marvin Edeas. He's using this advanced delivery system, which is the name to the nanoencapsulation method that he uses, And this has been used in these two products that you see here on the screen, which is Cronuit, which encapsulates Auxilia Night Reset, and Belage, which encapsulates Orzod enzyme 3 in this ADS. Dr. Marvin Edeas was postulated to a Nobel Prize in nanotechnology. His research focus is applied into the development of antioxidant technologies targeting mitochondria in the agro-food sector that are made from 100% natural products. He is the chairman of the board of the International Society of Antioxidants in Nutrition and Health. He is the founder and president of the French Society of Antioxidants, and he is the chairman of the board of several international congresses and conferences. One of the congresses that Dr. Marvin Edeas puts together is Targeting Mitochondria. The last edition of this congress was held in October 17 and 18 of 2013 in Berlin, Germany. This is the conference on diabetes and obesity. It is its 7th conference internationally. This was put together by the International Society of Antioxidants in Nutrition and Health in conjunction with Riga Strands University in Latvia. The 7th edition of this conference just took place in October 24 and 25th of 2013. This is the 3rd Congress on Reproduction, Oxidative Stress and Antioxidants in Fertility, Cancer and Obesity. This is going to be going on in May 22nd and 23rd of the year 2014 in Paris, France. It's also put together by Marvin Edeas, Dr. Marvin Edeas, in conjunction with the International Society of Antioxidants in Nutrition and Health. This other one is the Targeting Brain and Metabolic Syndrome, the Anti-Aging Challenges, and this has to do also with the International Society of Antioxidants in Nutrition and Health, and you can see the name of Dr. Marvin Edeas as the head of the scientific committee. This is the Congress on Skin Aging and Antioxidants, Innovations and Trends. This happened in October 29th and 30th of 2009 in Malta. This is the 6th edition of this Congress. It's also put together by the International Society of Antioxidants in Nutrition and Health and Dr. Marvin Edeas as the chairman of the board. Dr. Marvin Edeas is also linked to the World Stevia Organization, since they're using stevia in a lot of the formulations that they use with antioxidants. This is going to be the 6th World Congress on Stevia. It's going to be going on in June of 2014 in Berlin, Germany. On the previous edition of this Congress, you can see on the picture Dr. Marvin Edeas is giving an award to Dr. Per Bendix Jepsen from Aarhus University in Denmark, and this went on in 2013 in the month of May. This other Congress is on polyphenols applications. The 8th Congress is going to be going on in Lisbon, Portugal in June of 2014. You can see Dr. Marvin Edeas also as the head of the scientific committee of this Congress, in which also the International Society of Antioxidants in Nutrition and Health is involved. Dr. Marvin Edeas has published in PubMed.org 20 articles. Most of them are involved with the use of antioxidants. He also has extensive research in HIV. You can see a list of the articles here, and I'm going to show you one in the next slide. This is the article published by Dr. Marvin Edeas in PubMed.org. It was published on June of 2010. And it's titled Mylar Reaction, Mitochondria and Oxidative Stress, Potential Role of Antioxidants. And it says glycation and oxidative stress are two important processes known to play a key role in complications of many disease processes. Oxidative stress, either via increasing reactive oxygen species or by depleting the antioxidants, may modulate the genesis of early glycated proteins in vivo. Mylar reactions occur in vivo as well as in vitro and are associated with the chronic complications of diabetes, aging, and age-related diseases. Hyperglycemia causes the auto-oxidation of glucose, glycation of proteins, and the activations of polyol metabolism. These changes facilitate the generation of reactive oxygen species and decrease the activity of antioxidant enzymes such as copper and zinc cofactor superoxide demustase, resulting in remarkable increase of oxidative stress. A large body of evidence indicates that mitochondrial alteration is involved and plays a central role in various oxidative stress-related diseases. The damaged mitochondria produce more reactive oxygen species, which increases oxidative stress and less ATP or cellular energy than normal mitochondria. As they are damaged, they cannot burn or use glucose or lipids and cannot provide cell further with ATP. Further, glucose, amino acid, and lipid will not correctly be used and will accumulate outside the mitochondria. They will undergo more glycation, as observed in diabetes, obesity, HIV, and lipodystrophia. The objective of this paper is to discuss how to stop the vicious circle established between oxidative stress, Maillard reaction, and mitochondria. The potential application of some antioxidants to reduce glycation phenomenon and to increase the antioxidant defense system by targeting mitochondria will be discussed. Food and pharmaceutical companies share the same challenge and must act now urgently and energetically. Now I'm going to introduce you to Dr. Marvin Ideas' brother. His name is Dr. Bejit Ideas. Bejit Ideas is the President and Chief Executive Officer of Nina Farm in Japan. He's also the President of the International Society of Antioxidants in Japan. He's the Scientific Director of the Louis Pasteur Institute in Japan. He's a member of the American Anti-Aging Society. He's the Chief Innovation Officer of Sanki Major and a renowned international speaker in the field of nutrition. He has 2 PhDs, one in nutrition from University of Paris and one in biotechnology from Nancy University in France. Together and with collaboration of the institutions they lead and direct, they have developed the most advanced antioxidant active ingredients. They have also developed a patented nano-encapsulation technology by which the active ingredients are delivered inside mitochondria. And let's talk a little bit about the nano-encapsulation. And before I do that, I would like to explain the meaning of the word nano. Nano comes from nanometer, which is a unit of measurement of the metric system. And 1 nano is 1/1000th of a meter, or 1/1,000,000th of a millimeter. For comparison, I would like to mention that cells measure in microns or micrometers, and 1 nano is 1,000th part of a micron. This nanoencapsulation is a nanosphere that measures about 5 to 9 nanometers that allows the release of our active ingredients inside mitochondria. This nanoencapsulation is called Advanced Delivery System, and it serves several functions. It protects the active ingredients from degradation from the environment. It also protects the active ingredients from gastric acids in the stomach. It improves and activates intestinal absorption and allows the particles to enter into the bloodstream still intact. Once in our circulatory system, they are delivered to every cell of our body and into the mitochondria, where the active ingredients are released. The nanoencapsulation has amphiphilic and amphipathic properties, which allows them to swim through both water and fats. They swim through the blood plasma, which is 92% water, and then go through the cellular membranes, which are fat-based. Once inside the cell, they swim through water, and lastly, they go through the double membrane of the mitochondria, where the active ingredients are released and utilized. This happens 25 to 30 minutes after being ingested. Evidence of these amphiphilic properties of this advanced delivery system nanoencapsulation has been verified by Photonics Healthcare, who use cutting-edge technology to monitor oxygen levels inside mitochondria and increased function. This year, the Scientific Committee of Targeting Mitochondria gave an award to Dr. Flor Harms for her work utilizing Photonics Healthcare techniques which allow for monitoring mitochondrial oxygen utilization. In vivo, mitochondrial respirometry allows for measurement of mitochondrial oxygen tension and mitochondrial oxygen consumption in skin. These techniques are now being used in humans for the first time. This technology is also being used to measure the effects of therapeutics to target mitochondria. This technology was researched and developed by Dr. Bert Milk from Amsterdam and was presented at the Targeting Mitochondria in 2011. The first product that was developed by these scientists is an active ingredient called Orizod, which is a mixture of rosemary extract and olive leaf extract. It took a 15-year study where more than 50 antioxidant ingredients were tested, including green tea, resveratrol, and many others. But the synergistic effects of these two extracts proved to be the most efficient when being delivered with nanotechnology. Orizod, by the way, means olive and rosemary increment superoxide dismutase. Orizod was evaluated by a scientific group who specializes in antioxidants called Probiox Laboratories in Belgium. They are recognized as a world leader in oxidative stress management, and the company owns key intellectual property related to the analysis and interpretation of oxidative stress parameters. They are based at the University of Liège in Belgium, and they were established in 2001. This is the group of doctors that evaluated Orizod. They are all members of Probiox. They are the scientific committee, and they are led by Dr. Luc Montagnier, who was awarded a Nobel Prize in Medicine in 2008. Here you can see his award in nobelprize.org. He won this award for the discovery of the HIV virus, which is also a field very well known by Dr. Marvin Edeas, who has done extensive research in this field as well. This is an example of the research work that Probiox does. It was also published in PubMed. It is about the oxidative damage experienced by women who had taken oral contraceptives. In this study, significant increases in the mean levels of lipid peroxides and oxidized LDLs were observed in these women, increasing the risk for cardiovascular disease. By the way, lipid peroxidation is the main marker used to recognize oxidative stress. These are the results that Probiox Laboratories reported regarding Balash and its active ingredient Orizod. This study was conducted on 30 people, men and women, during 30 days in April and May of 2010. After taking Balash with Orizod for 30 days, they found a 21% increase in superoxide dismutase production, an 18% increase in glutathione peroxide production, a 38% decrease in lipid peroxidation pool, and a 25% reduction in oxidation of the low-density cholesterol. Since then, a newer version of Orizod was developed, and at the bottom of the screen you can see the results for this newer version of Orizod. It's now called Orizod Enzyme 3. This study was also done in 30 people, but it lasted 90 days. It ran from the beginning of June to the end of August in 2012. This second study was done by Medica Tokyo on Orizod Enzyme 3, and it reported that after taking Balash for 90 days, they observed a 25% increase in superoxide dismutase production, a 19% increase of glutathione peroxide production, a 43% decrease in lipid peroxidation pool, and a 29% reduction in oxidation of the low-density cholesterol, or LDLs. There are 20 years of research behind the development of Orizod Enzyme 3. It is not only the most effective active ingredient to combat oxidative stress, but its advanced nanoencapsulation allows it to reach the target, which are the mitochondria, where free radicals are produced. This bioactive molecule contains polyphenol-type antioxidants and superoxide dismutase extracted from plants which are grown in an ideal environment with the right sun exposure and fresh air in Alicante, Spain, and Toulouse, France. They are cultivated in private farms next to the processing plants, where the extracts are purified and nanoencapsulated in ADS to achieve the highest level of efficacy in the human body. Belash is included in the medical reference books in Mexico, where it was launched to market with this name. It is listed as an antioxidant supplement used to diminish the effects of oxidative stress and useful to prevent premature aging. The description of Belash inside the PR Vademecum explains: recent clinical studies demonstrate that the consumption of Belash stimulate and increment the activity of the two main enzymatic antioxidants SOD and GPX, which play a crucial role in the protection of cells against free radical damage. Long-term oxidative stress is considered the main cause for decreased cognitive function commonly observed with aging. Oxidation happens at the cellular level, and it's responsible for the deterioration and premature aging when it overwhelms the mechanistic limits established by antioxidants. When this happens, a high production of free radicals is produced, characterized by an elevated activity and the capacity to form more free radicals by a chemical chain reaction. The excess of reactive oxygen species promotes the attack over chemical compounds inside the cells, like lipids, proteins, and DNA. This triggers an initiation of a series of biochemical events that could lead to the appearance of physiopathological disorders, worsening of illnesses, and also alter the physical and or mental performance of a supposedly healthy person. It has been demonstrated scientifically that oxidative stress is the cause or consequence of over 250 diseases. Recent clinical studies show an increment that orisod enzyme 3 generates in the main antioxidant systems superoxide dismutase, glutathione peroxidase, and catalase, boosting the antioxidant defense capacity of the body. In addition, it reduces significantly the lipid peroxidation and low-density cholesterol oxidation levels. It supports the brain in production of energy, supports liver detoxification, and shows a decrease in free radical production. Belash and its active ingredient orisod enzyme 3 are appropriate to fight free radicals, especially when the body's own defense systems are weak, like older people, sun exposure, tobacco, stress, intense physical exercise, and chronic and degenerative diseases, among others. Belash can prevent some chronic diseases, including oxidative stress, reduce their development, and create a better quality of life for the patient. This is a slide that has been extracted from the PR Vademecum internet site. Here you can find all the details of the product and the most recent clinical studies made by Medica Tokyo. You can find a link of the page at the bottom of the screen. You will find the nutritional information, the ingredients, the therapeutic indications, the pharmacokinetic description, and the antioxidant activity found in the clinical study. This book, like most medical reference books, are country specific. So I am going to translate for you the next few slides since the information here is in Spanish. The results to the clinical studies demonstrate that orisod enzyme 3, the main ingredient in Belash, increases the body's antioxidant defenses, activating the SOD molecule. This way, it can preserve the enzyme's function and biological properties. After the consumption for 90 days, SOD increases significantly. The results to the clinical study also demonstrate that taking orisod enzyme 3, the main active ingredient in Belash, after 90 days, it stimulates the activity of glutathione peroxide enzyme, lowering free radical formation. Glutathione peroxide provides a detoxification mechanism of peroxides in live cells. This reaction has a crucial role in the protection of our cells against free radical damage created by decomposition of peroxides. The lipid components of our cells are prone to react with free radicals, resulting in lipid peroxidation. Glutathione peroxidase transform peroxides in alcohol using glutathione, preventing the formation of free radicals. The results of the clinical study demonstrate that taking Orisod enzyme 3, the active ingredient in Belage for 90 days, reduces the lipid peroxidation and free radical formation, lowering oxidative stress levels. Since polyunsaturated fatty acids are easy targets for oxidants, once the lipid peroxidation process has started, it produces a chain reaction in free radical production. Lipid peroxidation is the most common biomarker of oxidative stress. This slide is from the study done by Probiox Laboratories, so you can see the results. Even though they're in Spanish, most scientific words easily translate. These results are for the earlier version of Belage when it contained Orisod, which was the first generation of the product and active ingredient. This other slide is the study done by Medica Tokyo with the newest version of Belage with its new active ingredient Orisod enzyme 3. This was done in August of 2002, and I just wanted to mention that Orisod, or the letters in Orisod, means O for olive, R for rosemary, I for increments, and SOD. Orisod means olive and rosemary increment, SOD. Now let's talk about a product called Cronwheat. Cronwheat's active ingredient is called oxylianide reset. It is also nano-encapsulated with ADS, or Advanced Delivery System. It comes in powder form and it has a berry flavor. It can be diluted in either hot water or cold water for consumption. Oxilia was developed by Nina Farm in collaboration of the French and Japanese teams. Oxilia is a natural diet supplement with active efficiency on weight control and powerful antioxidant properties for anti-aging. It prevents from oxidation thanks to SOD effects and improves the metabolism. It also blocks calorie intake and prevents the storage of fat. Oxilia has been the best-selling product in Europe since 2005. It won awards as the best slimming product by the French Society of Antioxidants and as the best active ingredient to combat metabolic syndrome. Nina Farm, the developer of these active ingredients, does not market directly to consumers. Their focus is in the research and development of the active ingredients alone. So what they do is they partner up with other laboratories to send products to market. So in Europe, they partner up with Forte Pharma to market Oxilia Total, which was an earlier version of Oxylianide Reset. And this box you see here on the screen can be found throughout Europe, and you can find it printed in many different languages. And last year alone, in 2012, this box you see here on the screen, it sold 14 million times. Cronwheat can also be found in the PR Vademecum medical reference book in Mexico, where it is described as a preventive antioxidant to reduce the effects of oxidative stress. It also says that it is useful to prevent premature aging, to prevent or ameliorate metabolic syndrome and its consequences, and it's also a weight management and stabilizer. Cronwheat has four main benefits. Number one, oxylianide reset inhibits the activity of the enzyme alpha amylase during digestion and regulates the absorption of sugars and carbohydrates. Number two, the anthocyanins and proanthocyanins in the black bean shell have demonstrated strong anti-glycemic properties that reduce sugar and insulin levels after meals. 3. Insulin resistance. The antioxidants in oxylianide reset protect the pancreatic beta cells from oxidative stress induced by glucose. Number 4. Energy source. The olive and rosemary extracts promote mitochondrial function, and the manganese from the black beans is essential to reduce the effects of free radicals. This is Cronwheat in the PR Vademecum medical reference book in the Mexican site. You can find a link to the page at the bottom of the screen. It is listed as a natural supplement, and its active ingredients are black bean extract, olive leaf extract, and rosemary extract. It is a preventive antioxidant used to lower the effects of oxidative stress. It is useful to prevent premature aging. It prevents or improves metabolic syndrome and its consequences. It improves and stabilizes body weight. The study which results are published here are aimed to evaluate the efficacy of Auxilia Night Reset in several biological functions like diabetes, glycemic index reduction, body weight control, LDL and HDL balance, and the presence of antioxidants in the blood serum. The study was done in 30 people, men and women between the ages of 27 to 71 with a median age of 55, that were all overweight and had a large belly, which is an indicator of diabetes and cardiovascular risks. They were broken into two groups, and half of them were given Auxilia Night Reset and the other half took a placebo. They were taking physical measurements before treatment and 30 days into the treatment for their weight, corporal mass index, waist circumference, and hip circumference. Biological level analysis was also done before and after the 30-day treatment, and these were insulinemia, glycemia, HOMA index, which is an index of insulin resistance, total cholesterol, low density level cholesterol, high density level cholesterol, triglycerides, and antioxidant activities. These individuals were also chosen with high levels of glycemia postprandial, which means after eating, which is an indicator of early stages of glucose intolerance and which usually leads to type 2 diabetes. Insulinemia when fasting is also an indicator of early stages of insulin resistance. To evaluate the impact of Auxilia Night Reset to the glycemic response, a sample meal was given to all individuals to be able to get all of their biological measurements. The first part of the test was to compare the two groups' response score at given times after being given the test food. The efficacy of Auxilia Night Reset in the glycemic response to the test food was evident. We can see that Auxilia scores are lower than the control group related to the glycemic response. Auxilia's line, by the way, is here the bottom one, and you can see lower scores at all the given times that it was given, and up here you can see the group that took the placebo, all higher scores. This table shows the results for both groups during the test regarding their weight and corporal mass. The lines at the top represent the group that took the placebo, and the ones at the bottom is the group that took Auxilia Night Reset. And the table shows that both groups lost weight during the test, but it is much more significant in the Auxilia group. The first column refers to body weight at day 0 or before the treatment. The second column refers to the body weight after 30 days, and the third column shows the difference between the first and the second columns. The results are shown in kilos, so results show that the placebo group lost 0.31 kilos, which is 0.68 pounds. The Auxilia group, however, lost 2.26 kilos, which is very close to 5 pounds. This table shows a before-after as well as a difference column for waist, hips measurements. The placebo group shows a small increment in measurements, and the Auxilia Night Reset group show a reduction in measurements. These measurements are given in centimeters. On column 1 we can see the waist measurement at day 0 or before the treatment. The second column is the waist measurement at day 30, and the third column is the difference between column 1 and 2. The next 3 columns are the ones related to hip measurements, shown in the same way, before, after, and difference, and the last 3 columns are a ratio between waist and hips. The results in column 3 show that people in the Auxilia group reduced an average of 1.48 centimeters at the waist, and in column 6 it shows that they reduced 1.79 centimeters at the hips on average. The placebo group had negative results. having increased measurements. These results show evidence of the efficacy of oxygenate reset in weight, waist, and hips measurements in people that kept their eating habits. This table shows the results related to glycemia on the first three columns, in the insulinemia on the next three, and for insulin resistance on the last three. After a month, both groups had a reduction in glycemia. The Auxilia group reported a higher reduction in insulinemia than the placebo group. And lastly, the Auxilia group had a higher reduction of HOMA index of insulin resistance than the placebo group did. This table is for the metabolism of lipids, and it has results for triglycerides, total cholesterol, low density level cholesterol, and high density level cholesterol. We can see a higher reduction in all of the lipid measurements in the Auxilia group than in the placebo group. The first three columns are for triglycerides, before, after, and the difference. The next three columns are for total cholesterol, before, after, and the difference. The next three columns are before, after, and difference for high density level cholesterol. And the last three columns are before, after, and difference for low density level cholesterol. So if you just focus on columns 3, 6, 9, and 12, you can see that all the averages on the difference on each of them, and you can see a much higher reduction in the Auxilia group. Sanki is the company that is marketing these two products in America. Sanki got started in Mexico in 2010, and in 3 years it grew to almost 200,000 distributors and consumers. Sanki, now called Sanki Global, opened for business in the United States in June of 2013, with offices in Las Vegas, Nevada. The company is led by Alejandro Lopez-Tello, a chemical engineer and an entrepreneur with an extensive success record in network marketing. The company's headquarters are in Mexico City, and there are also offices in Lyon, France, and in Tokyo, Japan. The company was founded when Lopez-Tello met with Dr. Vegeta Diaz in Tokyo. Lopez-Tello was invited to market Nina Farm's active ingredients in Mexico, but he always wanted to give people the opportunity to grow with the company and prosper, so he chose network marketing as a business model. He partnered up with NC and Gerd Mueller, and they got started by working and designing an attractive compensation plan, and 3 months later they had reached break-even point, and before 6 months they were generating profits. A year later, Sanki signed an agreement with Major Laboratories, which is a cluster of Nina Farm in charge of all their in vivo research and testings focused in natural active ingredients. As part of this agreement, it was agreed that 10% of Sanki's profits would go to the research group now called Sanki Major Innovation Lab, to continue the research and development of new and existing products. Sanki Major is based in France and Japan, and it counts with a cutting-edge scientific group formed by doctors, biochemists, and formulators, supported by the most important scientific institutions around the world in the fields of nanotechnology, oxidative stress, anti-aging medicine, and mitochondrial therapeutics. By 2012, Sanki launched its second product to market. Cronuit is practically a need for the Mexican people, since the Mexican population is ranked as number 2 in obesity in the world. This same year, Sanki was recognized as a great place to work in Mexico, ranking number 11 in its category. This recognition was also achieved in the year 2013, and it is awarded by a global company focused on great workplaces who have the expertise and tools to help businesses transform and prosper through the leadership, development, and workplace culture consulting services. This is in fact the very first time that a network marketing company has ever achieved this recognition. The United States is the platform for a global expansion, and there are plans to be in all of the American continent in the next few years. Sanki Major is a global health and wellness laboratory dedicated to improve the quality of life by offering medicalized solutions based on a combined expertise of nanotechnology, nutraceutical formulation, and clinical testing. Years of experience with human clinical trials on metabolic syndrome, diabetes, obesity, and inflammatory disorders have enabled Sanki Major to create innovative medicalized solutions based on an in vivo expertise and an advanced knowledge in fermentation technology. Obesity and metabolic syndrome are the main health challenges for millions of people around the world, with more and more cases diagnosed every year. This slow-progressing syndrome demands fast-acting solutions as it has long-term devastating impact on health, like diabetes, cardiovascular disease, hypertension, etc. Right from its origin, Sanki Major has been present in the world's most important trade events related to health. Sanki Major has participated in 2 Vita-Food events, the most important global event on nutraceuticals, raw materials, functional food, and drink ingredients. Sanki Major was also present at the first World Summit on Medicalized Food and Medical Nutrition held in Paris in 2012, and in collaboration with the International Society of Antioxidants in Nutrition and Health, Sanki hosted its 4th Innovation Convention in Paris in May of 2013. Research is the foundation of our business, and we collaborate with a global network of scientists to develop natural and highly effective ingredients for nutraceutical applications. Sanki Major has developed its own functional systems dedicated to the synthesis and manipulation of molecules and nanomaterials. Innovations and products are all tested in vivo, from absorption studies to human in vivo clinical trials. The company was the first in the nutrition field to start in vivo trials for all of its formulas. Clinical expertise combined with the in vivo testing platform offers Sanki Major a strategic position to develop and share its innovations and medicalized solutions targeting obesity and metabolic syndrome. Sanki products have also earned the kosher seal from the Jewish community. You can see the certificate here. To earn this certificate, a rabbi is sent out to do a very strict and thorough inspection of the facilities where products are produced. He checks out every part of the process of production and inspects every piece of the equipment that is being used. The kosher certificate is a guarantee of purity, quality, and safety. Sanki was founded around a strategy called the Blue Ocean Strategy, which is the title to a book with the same name. This book was published by the Harvard Business School Press and refers to a strategy in business based in value created by innovation. The key to this strategy is to market products where there is no competition for market share. Innovation is what creates value and gives companies and the distributors a competitive advantage in the market. In contrast, in a red ocean strategy, companies are in a cutthroat competition for market share. Sanki gets its innovation from patented nanotechnology that has created a brand new category of nutraceuticals which have no competition. This has created a blue ocean environment for our distributors, giving them a constant competitive advantage and marketing edge. The United States, just like in Mexico, this is a huge opportunity since they are the 2 countries with the biggest demand. Health statistics show huge numbers in cases of obesity, diabetes, metabolic syndrome, and many other neurodegenerative, metabolic, and chronic degenerative diseases, where oxidative stress and mitochondrial dysfunction play an important role. This Blue Ocean strategy is also the cornerstone of our research and development team, who have a track record of Blue Ocean developments and new product pipeline. No other nutraceutical company competes in this category because they simply don't have this technology. No other product in the marketplace has the capacity to reach mitochondrial. I invite you to look into this amazing opportunity with Sanki, which is based in these 2 extraordinary products I just described. It is also based in the Blue Ocean marketing strategy, which is the backbone of this opportunity, where you will find no competition since there's no other nanotechnology bioactive ingredient able to act inside mitochondria. If you're interested in health, here you have the most effective way to fight free radicals which steal energy from us and make us age prematurely and lead us to all sorts of complications. We're creating a leadership and information platform to help you succeed with Sanki. If you've never been in network marketing before, I'd like to tell you that it offers unlimited potential and that we're projecting to be in the American continent in the whole American continent in the next 6 to 10 years. This opportunity is even bigger here in the United States, that under the direction of Jeffrey Boyle, who has an amazing track record in success in network marketing and has done consulting for over 20 other companies, helping them to launch internationally. He brings new tools to help you grow your business, adding to the ones that Sanki already had in Mexico. Register with Sanki and give yourself the opportunity to get to know the whole concept, the resources, and the compensation plan. We're continuously developing new tools and resources to help you succeed in the Sanki business if it is your desire. If you're only interested in getting product for yourself, a $35 fee will sign you up to get wholesale pricing. Those $35 will also give you the opportunity to refer others and grow your business. If you see an opportunity here with Sanki, we're here to support you and to help you grow in your business. Copy the links on the next slide and share this information with your loved ones. You will find health and wellness information to support your Sanki business. Help this amazing scientific group to continue developing solutions that work inside mitochondria, which are our energy factories. Help your cells boost their defense system. Help your loved ones have a better quality of life. Thank you so much for listening. I'll leave you on the next page with the links.
Conquistar, evolucionar, voy a tomar mi lugar en el mundo. Hay mucho más en lo que se puede ver. Soy un gigante, la cima voy a tomar. Y sé que muchos me acompañarán. Y yo, Sanki y yo. Estamos juntos, seremos la fuerza que mueva todo, que mueva todo. Seremos uno, una gran familia, la misma visión, la misma visión. Voy a decir, pero vivir mis sueños aquí. Somos aquí. Este es el lugar en donde yo quiero estar. Somos Sanki. Yo el camino elijo, mi destino. Tengo la fuerza para transformar. Voy a tomar mi lugar en el mundo. Hay mucho más en lo que se puede ver. Soy un gigante, la cima voy a tomar. Y sé que muchos me acompañarán. Y yo, Sanki y yo. Estamos juntos, seremos la fuerza que mueva todo, que mueva todo. Seremos uno, una gran familia, la misma visión, la misma visión. Voy a decir, puedo vivir mis sueños aquí. Somos aquí. Este es el lugar en donde yo quiero estar. Somos Sanki.